Alright, so now that we know a bit about DNA, let's talk about a central theme in biology. It's so important that they call it the central dogma of molecular biology. And that is that DNA moves to RNA and RNA goes to protein. First, it's important to know that DNA has a very specific structure. Remember the double helix and the 4 nucleotides that make up DNA? This allows it to copy itself in a process called DNA replication. This process is used for a number of things, including growth, repair, and reproduction. Now, the process of DNA going to RNA is called transcription. This is very similar to making a photocopy of a recipe out of a cookbook. And RNA moving to protein is a process that's called translation. So reading the copy that was made and making the protein out of it. And remember that proteins, long strings of amino acids, perform specific jobs within the cell. As mentioned, the structure of DNA allows for DNA replication. And that's because the nucleotides pair together in very predictable ways. So we can see that the thymine always binds to the adenine residue, and the guanine always binds with cytosine. Now let's practice. You have one DNA strand here. What would be the complementary strand? And remember our base pairing rules. So A always goes with T, and G always goes with C. So what would we have at our first position here? Our complementary strand would have a T, right? So A pairs with T. And how about this second thymine residue? It binds with A. T binds with A. C, remember, always binds with G. C, T, A, until we move down the line, and we should have something like this at our complementary strand. So as mentioned previously, DNA is copied in a process called replication, where one strand of DNA serves as a template for copying using this complementary base pairing rule. You can watch videos of this process more in detail in English and Spanish, and the links I've included here. And these are in the PowerPoint that you can download from Canvas. You might be asking, why do cells need a mechanism to replicate their DNA? So remember that DNA provides the instruction manual for life. So all new cells need their own DNA. So every time a cell divides that we can see over to the right, whether it be for growth, repair, or even reproduction, cells need to replicate their DNA. In this case, asexual reproduction would be mitosis. So we see an example of mitosis here where we have one cell splitting into two. So remember that mitosis creates two identical daughter cells. In the case of sexual reproduction, it would be meiosis, where we're going to cover it in a few weeks. Oops. But even if a cell is not actively dividing, it still is in the process of using DNA to make those proteins that have such vital functions to our cells. And this is through the process of transcribing that DNA to RNA, transcription, and translating the resultant RNA to protein, translation. Okay, now getting to the actual process of creating proteins, cells are always creating different proteins to perform all of the different functions that our cells have. Remember that proteins are the molecules that perform specific functions in the cellular environment. Now, the first step in making a protein from our DNA is the DNA needs to be transcribed into RNA or copied over through a process called transcription. The main purpose of transcription is to copy the DNA into an RNA molecule that can be transported out of the nucleus where the DNA lives to the cytoplasm, where it is then translated into a functioning protein. And you can see more details about transcription in the YouTube videos linked in this slide. I've included them in both English and Spanish for convenience. After transcription, mRNA, or messenger RNA, is read and translated from the nucleotides to the amino acids, similar to translating one language to another. Translation takes one language, the DNA and RNA building blocks, the nucleotides, and translates it into a string of amino acids. And a long string of amino acids is what makes up a protein. The mRNA code is read in groups of 3 nucleotides called a codon that you can see down at the bottom in our figure. So AUG forms one codon, and this codes for the amino acid methionine. Similarly, AUC, the next codon, translates to isoleucine, and UCG for serine. Each codon signals a specific amino acid to be added to the chain. And you can see more details about the translation process in English and Spanish in the links provided. Now, why are these proteins important? What do they actually do for our bodies? And the answer is a whole heck of a lot. So, there are proteins that pretty much do every function that we can think of in the cellular environment. So this includes proteins that primarily function to transport important molecules, like hemoglobin that we're going to learn about when we talk about the respiratory system and the circulatory system, certain hormones that regulate specific physiological processes, and even some that protect you from illness and disease in the case of antibodies. Now, you might be wondering, what happens if we change one of these nucleotides in the DNA code? Now, these changes are referred to as genetic mutations, and the answer is that it can cause some pretty drastic consequences for us. Think of translating one language to another. If you had one or a few letters that were different, it could make for a very confusing and difficult translation. There are 3 main types of mutations. One is what's called a nonsense mutation. This occurs if a stop codon is created in the middle of the mRNA strand. So remember that each codon codes for a specific amino acid, and there's a specific type of codon that codes for a stop signal, telling the translation process to stop. And so a nonsense mutation is when a nucleotide change occurs that actually causes the translation to a stop codon. Now, a point mutation occurs when a nucleotide is replaced with an incorrect nucleotide. Now, this can sometimes have very drastic consequences for us humans. Sometimes, also, there are no consequences. An example of drastic consequences is the disease sickle cell anemia, which is caused by a single base pair change in a specific gene related to the protein hemoglobin. And so red blood cells are shaped a very specific way, and that's because they need to squeeze into tiny capillaries and veins to take all the blood to the necessary areas of the body. But in the case of sickle cell anemia, the protein that carries oxygen to all the areas of our body actually misfolds, and it actually causes a different type of shape of the red blood cell. And this actually works to block blood flow to all of our tiny capillaries and veins. And so it can have quite a drastic effect on the individual. And last but not least, we have frameshift mutations. And this occurs when one or more nucleotides are inserted or removed from the DNA. And this can cause the genetic code to be misread during translation, resulting in a dysfunctional or non-functional protein. That concludes our DNA lecture, and thanks very much for listening. Just a few notes for this coming Wednesday. Any questions that you have from this lecture, we can discuss the first 10 to 15 minutes of our class. I've also put up discussion number 2, and this has to do with gene editing. And you'll have access to this discussion until February 5th. So that's not this Wednesday, but the following Wednesday to complete it. So remember, respond to the questions that are in the discussion and also reply to 2 others that have responded to the prompt as well. And at the end of last Wednesday's class, some very observant students noted that there is no quiz listed on the Canvas website. And so I will not be giving you a quiz this Wednesday. Instead, we'll play a game of Kahoot to make sure that you understand the concepts in this DNA lecture. But I do want you to study these exams, or not these exams, but this material, because it will be on your midterm exam. And remember to turn in the DNA extraction worksheet at the beginning of next lecture. And we'll work on our next activity. All right, thanks.